I want to express my deepest gratitude for the selection of glucagon-like peptide 1 as the subject for this year's Frontiers of Knowledge Award in Biology and Biomedicine. When we first pulled out this peptide from intestinal extracts and demonstrated its effects on insulin secretion, there was no way that we could predict the impact that this molecule has had on human health. We started by asking a very simple question. I came from surgery, and some of our patients developed hypoglycemia, low blood glucose. This only occurred after meal intake, and we thought that it might be due to an insulin-stimulating hormone from the gut, a so-called incretin. But these hormones were hypothetical in those days. So I found some gut cells that were producing a substance resembling glucagon. Glucagon is a hormone from the pancreas known to stimulate insulin secretion. Together with others, I managed to find out what this substance was. It is a peptide called glycensine, which has glucagon in the middle. We could also find the peptide in the pancreas, and we therefore proposed that it was the precursor of glucagon, pro-glucagon. Unfortunately, it didn't stimulate insulin secretion. However, it seemed that glycensine might only represent a small part of the full pro-glucagon molecule, so the hunt for the full structure continued, and in 1983, the molecular biologist Graham Bell in the US managed to clone the nucleotide sequences encoding pro-glucagon from hamster pancreases. His results confirmed that glycensine made up half of the precursor, but also showed that it contained 2 additional glucagon-like stretches. This was exactly the information we needed. We had the potential hamster peptides synthesized and raised antibodies against them, enabling us to create radioimmunoassays measuring GLP-1 and GLP-2. We were able to show that in the pancreas, pro-glucagon was not cleaved to produce the individual peptides, and the story might have stopped there. But in the gut, they were formed separately and were actually secreted in response to glucose. Unfortunately, Bell's GLPs didn't do anything to insulin secretion from our favorite tool, an isolated living pancreas from pigs. We therefore decided to search for the actual glucagon-like peptides in gut extracts using our radioimmunoassays. By biochemical isolation and purification, we could soon pull out the real GLP-1 from the extracts and determine its structure. It differed from the predicted structure and turned out to be a very powerful stimulator of insulin secretion from our pancreases. So here was a new insulin-stimulating hormone from the gut. Was it also the peptide that we were looking for? So several years later, we were able to prove using an antagonist of the GLP-1 receptor that GLP-1 was indeed responsible for the hypoglycemia that we saw in our patients. But what else could this peptide do? So we could show that it also inhibited the secretion of glucagon from the pancreas. And this was interesting because people with type 2 diabetes have too much glucagon. Because it was a gut hormone, we also looked at its effects on gastric acid secretion and pancreatic secretion, as well as gastric emptying, and GLP-1 inhibited all of these. And we could show that the actions actually involved the brain. Eventually, we could also show that GLP-1 inhibited appetite and food intake in people. All of this would be interesting for people with type 2 diabetes. And in 1993, we could show, together with Michael Nauk from Göttingen in Germany, that blood glucose was completely normalized in patients with severe type 2 diabetes during a 4-hour intravenous infusion of GLP-1. And in 2002, we could show that a 6-week-long subcutaneous continuous infusion of GLP-1 to people with long-standing type 2 diabetes had tremendous effects on blood glucose, insulin secretion, insulin sensitivity, and even body weight. We could also show that the new peptide almost evaporated from the circulation after injection, subcutaneous injection. Indeed, its half-life was only about 2 minutes. You cannot treat patients with a peptide like that. We also found out why this disappeared so quickly. It was broken down by an enzyme called DPP4, and we could demonstrate that breakdown could be prevented with inhibitors of the enzyme, and this would result in increased insulin secretion. That discovery resulted in the development of the very successful DPP4 inhibitors like sitagliptin, Januvia, used all over the globe for diabetes therapy since 2006. But with the effect of GLP-1 peptide in people with diabetes, we eventually managed to attract the attention of a company called Novo Nordisk. They decided to go for it and developed a long-acting GLP-1 analog similar to the long-acting insulins they had produced. It was called liraglutide or Victoza and had a half-life of 12 hours rather than 2 minutes. It was soon established as a superior anti-diabetic agent. A second-generation weekly version, semaglutide, was even more efficacious and caused weight losses of up to 18% in high doses. Sensationally, it also prevented complications and increased survival of the patients. Effective treatments for obesity and diabetes had finally been established.